Okay, so like we closed out last week and stuff was not working on some projects and working on others, and that's the nature of science. So we've got, we don't yet have any of our mutants purified, but we're in process of purifying mutant number 5, and hopefully it will be our first. We're also regrowing mutant 4, and we've got mutants 1, 2, and 3. We've got the plasmid back in the yeast cells, and we're trying to grow those. So, Lord willing, we'll make some progress on that front this week. We got some ancient DNA to sequence this week, so we've got some samples that Philly and Kenna prepped for us. So we're gonna get those ready today and tomorrow and get them sequencing. And so that's exciting. We're making more media and things like that. So we got a lot of different things that are kind of coming in here. The Basecalling model development is moving forward. We did some kind of sanity checks this morning, kind of looking at things, and we're realizing that it is working, but maybe there's some parts of it that are doing things that we hadn't thought about yet. So we're getting closer, making progress. This week we got some career interviews to do. We got some dinners with faculty and staff, and we also will make a trip. We're going to head to Dixon and to Nashville, make a couple different stops, and hopefully have some adventures along the way. So it's going to be a great week. So if we get it before we use it, we'll figure it out.
To prep mutant 5, so we're freezing down the little Yeti cup, or the cryo cup.
Do you want to pour it now?
We've got a
Do you want to pour it?
over suspended topo ready to put in it, and it'll freeze it down, and we'll be able to like crush it into a powder.
Setting up these little reaction mixtures to do a binding assay for this fluorescently tagged topo. So just diluting some DNA and then mixing up reaction mixtures, and we'll run them on the spec in a minute and see what happens.
Collecting fractions from our frac 30 so that we can get our topo out of them. Basically, this machine keeps track of a bunch of different factors, but what we're looking for is the UV to shoot up, because that's where our topo is when the UV is high. And so every time the frac 30 rotates, I take a fraction, and the ones that have the highest UV will take a sample and spec it in a protein gel to see if we have topo.
Okay.
It does a lot of different things. So it will take our sample, which is in this little tube over here, load it and run it through the pump, through usually the pressure sensor, but it doesn't work right now, through the wash valve, up to the injection valve, through this little column here, and then through the UV monitor, conductivity, and then it will run down into our fracture. Then it will wash twice. Once it will wash with just our wash buffer A, which is sitting up here, and then it will do, the second wash will be a rotation of our wash buffer and occasionally elution. It releases our topo, and we will collect it in these small purple tubes down here in the fracture. So once it's gotten to the elution phase, right now it's on its second wash, we'll switch from these clear tubes that I have into purple tubes, which are the smaller fractions, and that's where most of our topo should be. The topo is stuck in this little blue column. Once we elute it, it should drop down and drop into our tubes. So we'll have a lot more of it in this little column here. We've washed it with this P200 down here, and now we've put P750, which is a buffer, through the column, and it's going to elute it and allow the topo to drip into these tubes. And so once we get our tubes made, we will test them to see how concentrated they are, and then we'll take those concentrated fractions and we'll aliquot them into little tubes that we can use. Collecting fractions from our frac 30 so that we can get our topo out of them. This machine keeps track of a bunch of different factors, but what we're looking for is the UV to shoot up, because that's where our topo is when the UV is high. And so every time the frac 30 rotates, I take a fraction, and the ones that have the highest UV will take a sample and spec it in a protein gel to see if we have topo.
Okay.
Here's the picture spec. I'm spotting first P750 and then the other columns of topo that we got. What? James responded to my message.
So
There's stuff in the middle.
this dark band that we're looking at right here, that's our topo, and this is great news. It says we got it. We got to clean it up a little.
Yeah, we have topo.
It's like, wait, I got to record.
Continuing the it feels like ever growing march to getting this paper done. Right now what we're doing is we're trying to incorporate other sources. Basically we want to see what other people in the scientific community have said about some of the stuff we're doing. Because of that, we're having to make sure we cite all of our sources correctly. And currently we're trying to use this really cool program that allows us to do it automatically, but it's giving us some trouble, so we're figuring that out.
Okay, thank you.
No problem.
Well, try to think about it.
Which cannot be free.
Design. Well, right now we are harvesting some topo. So we just spun these through the centrifuge, and we've got us a little pellet right here. And so right now we're pouring off the liquid, and then we're going to put more yeast on top of that. Yeah.
Right now, I am setting up my drug dilution for a DNA cleavage assay for mutant 5. So, hello. Right now, I am running a relaxation enzyme titration. So, big fancy words, but I'm basically testing to see how active our mutant 5 topo is the more we dilute it. So I've got it as concentrated as it can be, all the way down to 1/40th of what it normally is, to see how active it is, see what we can do with it.
Ends up finding a good amount of phenocrylaceous. So it's like
University, where I fell in love with the career I'm currently in. It was the very first rotation I did my third year.
A Linux KVM system. So, you know, the industry you'll find is not near on the cutting edge, and it's not as shifting as much.
I tell you what I do. I throw away the cars.
Like shafts, like the little shaft rods that you put in like a line. Oh wow.
The Wonders of Creation Conference.
Wait, I need to share that picture with me, the one with the bear.
There, but I can't because I am staring straight into the sun. And but it's really good to have you here, and we're glad to be with you. Excited to get to join you today for this conference, and over the next few days. I brought a few of my students with me, and we're enjoying this together. My name is Joe DeWeese. I teach at Freed-Hardeman University in West Tennessee. I want to talk to you today a little bit about some projects we're working on that we actually do real science. And so this is kind of where grad school starts, med school starts, is down this hallway, or the big classroom. So we'll walk down this hall. I have no idea if they're in class or not, but we'll walk down this hall, and there's 2 or 3 big lecture halls along this hallway here. And one of them is usually grad students, and the other here since the fall of 24, pretty much.
Summer, yeah, I started out.
Okay. So it's where like the med school students learn, and then the library is the next building beyond that.
Yeah.
So,
Yeah.
So it's a whole row of buildings.
Yeah.
So we'll let
1, 2,
Joan
3, 4.
take us
Come
on down here.
Come on down.
All right, so we just spent some time touring. We went through Preston, Robinson Research Buildings, and then Light Hall. We toured through some labs, the Ostroff Lab and Bomic Lab, and then we were able to kind of walk by some of the classrooms of the med school, the grad school, and now we're going to make our way around the library and head back out. But we got to meet some researchers, some scientists, and just kind of learn about what they're doing a little bit and see the facilities. We are working on our ancient DNA project, where we're trying to teach the Basecalling algorithm how to identify things like uracil in the DNA. And we realized there's a lot more layers and nuances to this than we thought. So we're going to run some PCR where we add in uracil at fixed concentrations and see if we can detect that increase in uracil on the output side when we actually sequence this. So right now it's just a big setup for PCR, quite a few reactions. We're going to kind of do several different conditions with several different plasmids, just to try to get enough that once we sequence it, we can kind of tell what's going on. So that's where we're at. Here's my rule of thumb: if you're doing anything less than 10, use a 10.
Yeah.
Don't use a 20 on less than C, G, and here's U. And there's right of some type. Now there's different algorithms for this. Muscle, Kruskal-Wallis, there's a whole host of different series of alignment metrics that weight things differently. Like one model may prefer that this matches up, another model may prefer something else matches up. Okay, so these different models have their own strengths and weaknesses as far as what they prefer and how to align it up. But the idea is, you know, I want an alignment model that if I have that exact same sequence, it's gonna align my little fragment up with that sequence. Now, if for some reason my sequence doesn't match, let's say this was a G, okay? Sometimes what it will do is just align it like this and say, here's a mismatch, okay? If it thinks that there is like some kind of insertion here, like let's say my sequence said C, G, G, A, now all of a sudden it's gonna insert two blanks here into that consensus sequence and say, oh well, there's, you know, there's this, and then there's these two, and then this, the rest of this lines up. So it's trying to figure out how best do they fit, and if that means adding a base in somewhere and shifting everything down, it's going to do that. And when you do that across 1,000 sequences, everything's trying to, you know, iteratively separate out. And so maybe it's best not to try to do everything, right, but to pick a pool or a sample so that we don't get too much of that and still get to this. So stuff like that is coming out of these different data sets. This or disturbance in the frequency of bases that we're seeing in these. Now this is a little tricky.
Oh!
What are you doing?
I am running a binding assay. So I finally got the hang of one thing, and we said, you know what, we're gonna change it up. And so I think, from what I can tell from the procedure, we're basically testing how diluted up topo can be and how well it will bind to DNA.
Okay.
I think is the main point. And so right now, the way that this works is you kind of set it up just like a relaxation. So you do your master mix, you have your enzyme dilutions, but instead, when you get to put the master mix on, you put it on the temp block for about 6 minutes. And then after that 6 minutes, you load it directly into the gel with dye. You don't stop it, you don't wait for a certain amount of time to stop it, like it's just running in the gel. So it'll show us hopefully some good things. I'm not too sure yet.
All right.
Do segregate according to structural themes. Here I've highlighted them in color.
And they're made by the, I don't know. All right, so we are wrapping up our 5th week. We got 1 week to go. The last 2 weeks have been busy. They've been packed. We've purified some of our mutants. So we've got about 3 of them purified now. So we're really excited about that. We've also been able to start experimenting on those to see which ones are working and which ones aren't. Last week we also got to go to a conference for a few days and hear some great speakers and have a good time visiting the Wonder Center as well as Vanderbilt. Just had some fun interactions there and did an escape game in the middle of all of that, which was fun. Our Ancient DNA project is still advancing, still moving forward. We're dealing with data analysis and trying to test out Ghost Shark, which is our Basecalling model, and really validate it. And so we've got some data sets we're working on toward that. This week has just been more experiments, running things, getting things done. We've done some more interviews, and it's just been a lot of fun. So next week, to kind of wrap up, we're going to be looking at trying to purify another mutant, hopefully to grow another mutant up, and then run some experiments on the ones that we purified that we haven't been able to test yet. So we're looking forward to that. On the ancient DNA side, we got a meeting coming up with our collaborators where we're trying to talk through some of our data and just understand what we're looking at. So looking forward to it. It's going to be a great time. Thanks for being out there.
6, 7.